Hello, everyone. My name is Frank Fabella, and I am a master's student at Cal Poly San Luis Obispo, working in the Environmental Proteomics Lab with Dr. Lars Tomanek. Today, I'll be giving a talk on a physiology experiment I conducted as part of a larger study trying to integrate multiple levels of biological organization and understand how previous thermal history, food ration, and sirtuin activity allow organisms to withstand an acute heat shock. To get a better understanding of this study, I recommend you watch Dr. Melissa May's talk, also being given at this conference. Our lab focuses on responses to environmental stress within the marine environment, such as the intertidal zone. The intertidal zone is characterized by tidal cycles that change the environmental conditions, such as temperature, salinity, oxygen, and food availability. With increasing perturbations by climate change, this environment is expected to become increasingly stressful than it already is in the near future. One organism that has thrived so far in this environment is the marine mussel Mytilus. These mussels are ecological engineers and create a 3D scaffolding structure allowing other organisms to live in. Marine mussels have been used as a sentinel species to measure environmental change and have been used extensively to monitor environmental stress responses, making them a good target organism for our studies. But what is environmental stress? An environmental stressor is a change in the environment that causes a shift away from homeostasis, thus causing a detrimental effect within an organism. These effects may occur at the organ and cellular levels. To combat environmental stressors, organisms change their behavior, such as moving away to a more habitable area. However, mussels are sessile and unable to move away from stressors. Previous studies have shown mussels are able to combat oxygen deprivation by opening their shells during low tide to increase oxygen availability. However, most of the studies in the mussels in response to environmental change occur at the cellular level. Mussels increase activity of heat shock proteins, as well as oxidative stress proteins to combat environmental change. These proteins are involved with maintaining cellular structure and functionality as part of a larger stress response. This response requires additional input of energy over basal requirements, involving diversion of energy to reestablish homeostasis. These shifts in metabolism cause changes at the organ level depending on the intensity of the stress. Muscles have evolved to withstand cyclic stresses. However, as I mentioned earlier, environmental stressors are expected to increase in intensity and duration due to a changing climate. This warrants investigation into how muscles will respond and recover from more intense environmental stress in the future, what regulates these responses at the cellular and organ level, and how are the responses at different levels of organization related. In a study performed by Lindsay Fitzgerald de Hoog, she acclimated mussels to a high and low food ration, and within each of those groups, acclimated mussels to a 32-degree or a 20-degree aerial temperature. After acclimation, she exposed all mussels to a 36-degree acute heat shock. What is interesting is that the high food, high thermally acclimated treatment had the highest proportion of mussels surviving. Additionally, the low food, high thermally acclimated treatment had comparable portions of survival as the high food, low thermally acclimated treatment. What is perhaps most interesting is that the low food, low thermally acclimated treatment also had a proportion of mussels surviving, despite having a low energy budget and no previous thermal history. So how is this possible? One possible mechanism is the protein family Sirtuins. Sirtuins regulate proteins by deacylation, meaning they cleave acyl groups off of target proteins that increases the activity of that target protein. Sirtuins are NAD dependent, meaning when food and nutrient levels are high, sirtuin levels are low. Sirtuin activity is high when food and nutrient levels are low. Additionally, our lab has shown that sirtuins partially regulate proteins involved in metabolism and the cellular stress response. So we know food ration and previous thermal history are related. We also know sirtuins depend on food availability and partially regulate the cellular stress response. However, we do not know what effects sirtuin have at the organ level, nor do we know how these proteomic changes integrate with organ level responses under environmental stress condition. I set out to fill the gap in our understanding and question how does previous thermal history, food ration, as well as sirtuin activity affect recovery from an acute heat stress at the organ level. I hypothesized that increased thermal acclimation and increased food ration increase the ability of muscles to recover from an acute heat shock, shown by a shorter recovery time. Additionally, increased sirtuin activity also increases recovery ability. On the other hand, decreased sirtuin activity and a lower thermal acclimation decrease muscles' ability to recover from an acute heat stress shown by a longer recovery period. To do this study, we used a tidal simulator that mimicked tidal and circadian cycles, regulated water and air temperatures, timed and measured food rations, as well as maintained oxygen content. We acclimated muscles to 4 different tanks for 3 weeks. We had a high and low food ration, and within each of these groups, we acclimated muscles to either a 30-degree or a 20-degree aerial temperature. Within each tank, we had 2 baskets. One basket received a sirtuin inhibitor and one did not. Prior to sirtuin inhibitor exposure, we took measurements at every high tide period for 2 days to obtain a baseline measurement. We then exposed the designated muscles to a sirtuin inhibitor. Then, all muscles in the experiment were exposed to a 33-degree acute heat stress to simulate an extremely hot day during low tide. We then took measurements at every high tide period for 2 days to monitor recovery. For my study, measurements were taken on excised gill tissue, an organ with high metabolic activity and has been previously used as a measure for overall metabolic state of muscles. The gill is used for respiration as well as feeding, making it a good target organ to study in this experiment. Tissue was placed on a temperature-controlled slide, filmed, and then analyzed using software created to measure ciliary beat frequency. Ciliary beating mechanistically moves food towards the mouth and pieces out viable nutrients. Studying these actions may give insight to recovery from stress by ingesting nutrients and therefore increasing the organism's energy budget. In our results, we found a significant 3-way interaction between the acute stress, the tank acclimation, and the presence of an inhibitor. We also found the significant effect of acute heat stress and the time of day. However, I will not be talking about that in this talk today. Here, you can see all the treatment changes before and after the acute heat stress. On the columns, you can see the food groups, and the rows show the thermal acclimation. Ciliary beating was mass-corrected and log-transformed to show these trends. Recovery from the acute stress was measured by comparing ciliary beat frequency before the acute heat stress to after. Beating frequency after stress that was similar to prior levels showed adequate recovery. All the changes that you see here before and after the acute heat stress are not statistically significant, except for the 20-degree high food treatment, meaning it had a reduced ability to recover. All of these other treatments theoretically should have had some kind of mechanism to combat the acute heat stress, whether it be a high energy budget, a high thermal acclimation, or the low food treatments, which would have high sirtuin activity to regulate a cellular stress response. The 20-degree high food treatment, again, should have had low sirtuin activity due to the high nutrient levels and high energy budget. As you can see from the graph, the inhibited treatment had a significant decrease in ciliary beat frequency. We can also see that the non-inhibit group did not have a significant change after the acute heat shock. However, these two groups should have had more similar beat frequencies prior to the acute heat stress. Despite the difference between the treatments being statistically insignificant, we are investigating why this might have happened. However, the data does suggest inhibiting sirtuin activity decreases ciliary beat frequency in well-fed muscles with no thermal history. The non-inhibited group should have had a high energy budget but some sirtuin activity. The high energy budget would allow these muscles to allocate energy to mounting a stress response. However, the inhibited group also had a high energy budget, but with the presence of inhibitors, the lack of any sirtuin activity did not allow for a similar recovery in the non-inhibited group. So what does this all mean? As we saw in the previous graphs, muscles are generally able to recover ciliary beating within a few days following an acute heat shock. However, sirtuins, food ration, and previous thermal history interact to alter the specific responses. Specifically, sirtuin actions may be most important in muscles exposed to high food and low thermal acclimation seen by a decreased recovery of ciliary beating. Additionally, in other treatments, previous stress such as low food and high temperatures may increase the levels of constitutive stress proteins within cells, allowing a quicker recovery from an acute stress at the organ level. To gain more insight, we also collected data on particle transport. We measured the speed and velocity of particles moving across the gill as a measure of ciliary beat efficiency. Additionally, I am in the process of collecting proteomic data using mass spectrometry to measure changes in protein type and abundance in each of these treatments over time. Hopefully, I will be able to integrate the proteomic data to the organ level data we saw here. Then, I will be able to mechanistically determine how food ration, thermal acclimation, and sirtuin activity change cellular proteins to affect ciliary beating. This process has been difficult, and it is hard to understand the interactions that are occurring by just looking at one level of biological organization. Looking at multiple levels, we will gain a more holistic view of what is actually occurring in these organisms and be able to determine how these ecologically important populations will withstand increasing perturbations of climate change. I would like to thank the National Science Foundation, as well as the CSU Council on Ocean Affairs, Science and Technology for funding this project. Thank you to all who have helped make this project possible, especially Dr. Melissa May and Dr. Lars Tomanek for all the support they gave me throughout this project. Thank you.